All right, y'all, you ready? Week 5, let's go. We're going to review a few different things. We kind of hopped around a little bit, so this is what we're going to be going through. We're going to be going through the glands, endocrine versus exocrine, how they're different, how they're similar. We're also going to talk about the structure that helps its function, as always. We're going to talk a little bit about cell-to-cell connections. I'm going to use a sheet that I handed out in class. We're going to talk a little bit about tissue healing and how we recruit different things to come to the site of damage to help it heal. And then last, I'm going to talk about skin layers and burns, okay, and how they relate to the skin layers. So let's get started. First off, glands. Glands are basically collections of epithelial cells. So these are epithelial cells that secrete some form of fluid or another. Now, in the name, we can assume what's going on here. So for exocrine, exo means outside, and crine literally means either to cry in Latin, but I'm going to say to secrete. So it's basically crying tears of secretions. Crying tears of secretions. So in exocrine glands, the secretions that are made by these cells, these secretory cells, are eventually going to make their way out of the body. Okay, so let me show you a gland like that. This is one type of gland. There are several different orientations, but the biggest thing that you can see with exocrine glands especially is that they're very long and branched. This allows for increased surface area on the apical side, these top sides of the cells, to basically allow for more cells to pack into one space to increase the amount of secretions that they can make. Now, these are epithelial tissues, like I said. They are usually cuboidal epithelial cells or columnar. The reason being is because these are pretty thick, big cells that have room for organelles to actually make the secretions themselves, that you need organelles to make them. Yes. Just a second. So like I was saying, cuboidal and columnar have more space for organelles to make those secretions, and that's why they're bigger. If they were squamous cells, if they were all flat like this, there's like no room for organelles, there's no room for stuff to go on in those cells. So that's why they are columnar or cuboidal. In this case, it's going to be cuboidal. When they secrete their fluid, they're going to secrete it into this large opening, well, large in terms of their size, into their larger opening called the lumen. Lumen is just a name for a hollow tube or hole. So you have gut lumen, that's like the inside of the gut, that hollow tube, where all of these secretory cells are going to secrete their fluid into the lumen and eventually go out via a duct. That's the key indicator that it is an exocrine gland. It contains a duct that will then go out of the body. For example, we've got several different glands that have ducts. You've got your salivary glands that will eventually go into the hollow tube of your mouth, so technically out of your body. You can spit, you can swallow, but then the swallowing, it would go out of your body eventually. Salivary glands make saliva, so that would be the secretions. You can have sweat glands. So sweat glands, they would secrete the sweat into that area, go out through the duct, get on your skin, cool you off. We also talked about, I'm blanking for a second, give me a second. Sweat glands, sebaceous glands. These are very active during adolescence because you are secreting a lot of oils, and that's why you get acne sometimes, these overactive sebaceous glands. And then you can also think of tear ducts. Tear ducts, they generate tears, and it comes out of your eyes and your tear ducts. Okay? The other one I'm going to tell you about is the pancreatic fluid. So pancreatic fluid contains sodium bicarbonate. This is an exocrine gland because the sodium bicarbonate, as we talked about in previous videos, goes into your small intestine, your first part of it's the duodenum, and it's going to go in there and eventually make its way out of the body. But the reason it's secreting into that hollow opening is because it's neutralizing all the acid from your stomach. Okay, so since it is going out of the body eventually, it is considered exocrine, and that will have a duct. We'll see the pancreas again in the endocrine system, though, too. So that is the exocrine glands, their shape, their function, and some examples. Awesome. Endocrine. Endo means inside, And when we're talking about endocrine glands, they're going to be inside secreting into the blood. So that's the key for endocrine. If something's getting secreted directly into the bloodstream, as opposed to a duct going outside of the body, it is an endocrine function. It's an endocrine gland or an endocrine cell. So for these examples, we've got our cells. Let's say this is in your pituitary gland, so that would be a great example of an endocrine gland, pituitary. It's at the base of your skull. And it's going to secrete different secretions into the bloodstream. It usually does this by exocytosis or just by simple diffusion, but we'll say exocytosis so you can remember it. Remember the vesicles will be surrounding it, and it'll eventually fuse with the membrane, and now they will get into the interstitial space, eventually get into the bloodstream. So all of these are usually coined hormones, because hormones are basically chemical messengers carried in the blood. So endocrine functions always go into the bloodstream via hormones, and that is the secretions that those cells give out. So the big difference between exo and endo goes into the blood, exocrine goes out of the body through a duct. A couple other examples, the pituitary gland has a ton of hormones. The thyroid has a few, but it's regulated by the pituitary, so the pituitary is like the master regulator. And then I wanted to tell you about the pancreas, specifically pancreatic beta cells. These secrete insulin. And if you could guess, where does the insulin go? Into the bloodstream. So insulin is a hormone that goes into the bloodstream from the pancreatic beta cells, because insulin is going to kind of act as a key that opens the lock to the cells. So it opens the gates up in the cells to allow glucose to flow in. So when you have high blood sugar, you just ate a high carbohydrate meal, your blood sugar is going to raise, and you need all that glucose to get into your cells. So insulin is the little key that opens the lock, the ligand-gated channel, remember that, that allows glucose to flow into the cells. Thereby, that cell can now make ATP by breaking up the glucose. So that is exocrine versus endocrine. Endocrine glands, they have an entire system dedicated to it in anatomy and physiology too. I have a review video up for that if you'd like to watch it. Okay, so we got done with our glands, and now let's talk about these cell-to-cell connections. I'm not going to take too much time with these. You should have received this paper. You can also see it online. But there's several cell-to-cell adhesions that I want to talk to you about. First off, when we look at like a gland, like all these cells, it almost looks like they're sharing a membrane, but they're not. They usually are connected by some of these cell-to-cell connections. So imagine this little section in the middle is in between each and every one of the cells to keep them together, okay, for certain tissue types, right? So let's walk through them. Top one, tight junctions. You see the plasma membranes coming out a bit. Since the plasma membrane is hydrophobic, this is a water-tight junction. Water cannot pass through these tight junctions very well, nor can many things at all. The adherens junctions and the desmosomes both connect them through these integral proteins in the membrane, as well as these filaments that go inside the membrane themselves. So there's both an intracellular component and an extracellular component. The adherens, A, is connected by actin. This is a thinner filament, so it's more flexible, but it's less strong. Whereas the desmosomes, they have very thick intermediate filaments, and they're much stronger and more stable, okay? Gap junctions, as we move down, they actually share, it's like a tunnel between cells. They share both a plasma membrane and cytoplasm. And this allows, this is what I want you to write down, ions to flow quickly from one cell to another. So if this cell were to gain a bunch of positive ions, well, normally cells are negative. So if this negative cell over here is connected by a gap junction to a positive cell, now that has a lot of positive ions, those positive ions are going to flow through the gap junctions into the other cell. This happens in your heart a lot through intercalated discs. You may have heard of those in lab with your cardiac muscle. Lastly, you got hemidesmosomes, hemi meaning half, and desmosomes usually connect a cell to a cell, but a hemidesmosome connects a cell to the extracellular matrix. And this is the hemidesmosome. This is an actin-linked one. It's not a desmosome, but I'm only going to talk about the hemidesmosomes on a test. I wouldn't talk to you about this one, so don't worry about that. So those are different types of cell-to-cell connections. That's basically all I went through in class. And there's several others. I'm just kind of grouping them into small categories for you. All right, last couple things. Tissue healing. Look at my beautiful diagram over here. It's going to be hanging in an art show here very soon, sold for millions. Let's say you get a cut on your wrist, your carpal region. Okay? If you were to zoom in on that cut, you would see this opening that you just burst a bunch of cells open, and potentially if it's bleeding, you got into your dermal layer, which we'll talk about in the skin here in a second. So there's a blood supply, so you start bleeding. Squeaky wheel gets the grease. We want to fix the area that's broken, right? So we need to get some things to that area to heal it up. There are two things that I talked about that will go there. Macrophages, big eaters to basically clean up the area, eat any bacteria or viruses that might get into there. And then fibroblasts. Fibro meaning fiber, blast meaning to build. So it's going to build fibers to basically plug up this hole that we've just made into our bloodstream, to plug it up so that we don't bleed anymore. And that's also a platelet-mediated process, and we'll talk about that later on in AP2. So we need to get these guys to that site. Right now they're not there, they're over here. We need them to move to that area. What do we use? Chemotaxic agents. So in that area, there will be a release of chemotaxic agents that will kind of slowly move out like this, from high density to low density. And these chemotaxic agents, I'll draw in this color, chemotaxic agents or molecules. Chemo means chemical. Taxi means to move, right? If you get taxied around. So it's basically chemicals that induce cells to move towards them. So the cells will detect these chemotaxic molecules. And basically, just like climbing a ladder, they're going to follow those chemicals to the site of the breakage. It's a fascinating process. There's three main ones that I want you to remember by memory. There are three types. Histamines. You may have taken antihistamines. They help with the inflammation. So histamines bump up inflammation, the redness, so that you get a bunch of healing in one area. And antihistamines help when your nasal cavity specifically is inflamed from pollen, from allergens, and antihistamines kind of diminish that inflammatory response, so the swelling goes down and you feel better. So histamines are one. Those are a type of chemotaxic agent. Prostaglandins are another. Prostaglandins. And then lastly, leukotrienes. Leuko meaning white. So leukotrienes bring white blood cells to the area. And what's nice about your body is you'll have white blood cells kind of hanging out right underneath this epidermal layer. So they're just ready to roll. So they're ready to move where they need to go to fix the infection or to fight off the infection. I think that's all I went through in class, so I'm going to stick it there. So I use this analogy. The chemotaxic agents are like the Batman signal in the sky, and Batman are the macrophagic fibroblasts, right? Wherever that is, they're going to migrate to that area. Awesome. Last thing. This was very short in my Monday, Wednesday class. We talked about the layers of skin, and I need some more room. So if you look up here, there's three distinct layers of the skin. This is most superficial. This will be the epidermis. Epi meaning on top of. Therefore, this section is the dermis. So the epidermis is on top of the dermis. And then this is the subcutaneous tissue. You may hear it be called the hypodermis. I like that terminology because it's underneath the dermis. The epidermis is stratified squamous epithelial cells, so those flat layers of cells. The dermis contains like every type of tissue. You're going to have some simple cuboidal in your glands. You're going to have some dense irregular connective collagenous tissue under here, kind of keeping everything together. You're going to have areolar right underneath the epidermis. There's a bunch, right? There's a bunch of tissues in the dermis. So this is the big player right here. The subcutaneous is primarily adipose tissue, so fat cells, and that's going to connect to your muscles. Okay, so it's all connected. There's also 5 layers of the epidermis. So I'm going to label them from superficial to deep. And they're all prefaced by stratum. So if I say stratum something, that's one layer of the epidermis because stratum means layer. So the most superficial layer, actually, let me remind you, there's a great mnemonic. It's come, let's get sunburnt. Come, let's get sunburnt. So the first layer on the outside is the stratum corneum. The second layer is the stratum lucidum. Next one is granulosum. Next one is spinosum. And the last one, aptly named, is the basale, or the basal layer. Oops, basal layer. Good enough. So it kind of makes sense. The corneum is the outside, cornea is the outer part of your eyes. Lucidum is light. It's very clear. Lucid dreaming is clear. Granulosum, there's granules. Spinosum, I hate because there's nothing specific about them, but it's right above the basal layer. And as skin cells divide, so once again, this is just in the epidermal layer, as skin cells divide in the base, in the basal, the stratum basal, they're going to push upwards. So all of these cells are going to get pushed upwards eventually. And if they get pushed upwards, they're getting away from the blood supply that's in the dermis. If they get away from the blood supply, they'll lose their nutrients, therefore they will eventually die and slough off. Okay? They'll also become keratinized, which gives them a protein, a stronger barrier on the outside. So that would be keratinized, stratified squamous epithelial tissue. Lots of tissues here. Burns, last thing. You've got three types of burns. You got first degree, second degree, third degree. Third degree is the worst. First degree is, I guess, the best, even though no burn is good. Epidermal layer, if it burns through, first degree burn. Through the dermis, second degree burn. Third degree burn, through the subcutaneous, so very deep. Either second and third degree burns both require skin grafts, so basically putting skin on top of it, because you basically opened up your body. So if you cut here, you've got blood vessels in here. So now your body is open to the environment, ready for bacteria to just jump right into your bloodstream. So you need to cover that up with a skin graft in order to prevent infection. All right, hey, that's week 5, guys. Hope you learned a lot. Hope this was helpful. Drop any questions in the comments below. Like and subscribe. Maybe I'll give you extra credit for it. We'll see. Take care.